Good morning, ladies and gentlemen. Every successful journey reaches its destination with gratitude, reflection, and celebration. It gives us immense pleasure to welcome you all to the third and final day of Elevate 2026, the three-day international conference on synergizing artificial intelligence and classical research in biosciences, engineering, and material science. Over the past 2 days, we have witnessed inspiring keynote lectures, stimulating research presentations, meaningful discussions, and wonderful networking opportunities. On behalf of the organizing committee, we extend our heartfelt welcome to all our distinguished keynote speakers, respected dignitaries, faculty members, researchers, delegates, scholars, students, and participants. Today, we conclude this remarkable academic journey with 2 enriching plenary lectures, followed by the valedictory session, where we celebrate the achievements and contribution of every participant. We wish you all an inspiring and memorable final day. Moving on to the plenary lecture 7, we are honored to begin today's proceedings with our 7th plenary lecture. It is our privilege to invite Dr. V. Vijay Kumar, Professor, Department of Chemistry, School of Advanced Sciences, Vellore Institute of Technology, Vellore. He will deliver a lecture on modified Dormer hydrogen transfer and Suzuki-Maillard cross-coupling reactions in the synthesis of quinoline motifs. To introduce our chief guest, may I invite Mr. A. Ashokan, HOD, Faculty of Commerce, to deliver the chief guest introduction.
Warm good morning to one and all present here. I am very happy to introduce our today's esteemed guest, Dr. V. Vijay Kumar sir. He is a professor in the Department of Chemistry at the School of Advanced Sciences, Vellore Institute of Technology in Vellore, Tamil Nadu. Born in Tamil Nadu, he completed both his bachelor's and master's degree in chemistry at Madurai Kamarajar University in Madurai. He then earned his PhD in nitrogen heterocycles from Gandhigram Rural Institute, Deemed University. Prior to his current position, Dr. Vijay Kumar sir served as lecturer in the Department of Chemistry at Gandhigram University and later as a lecturer in chemistry at the School of Chemical and Biotechnology at Shastra University in Tanjavur. He has published over 250 articles in various international journals. His H-index is 23 in Scopus and 27 in Google Scholar with an I-10 index of 86 in Google Scholar. Under his guidance, 14 PhD scholars and 2 MPhil scholars have completed their degrees. And currently 6 students are pursuing their PhD under his supervision. Additionally, more than 50 MSc and MTech projects have been completed to date. Currently, his research group focuses on the synthesis of various heterocycles along with their biological applications and sensor behaviors. Ladies and gentlemen, with great pleasure and immense respect, let us extend a warm and heartfelt welcome to our esteemed guest, Dr. V. Vijay Kumar sir, with a big round of applause. Thank you.
Now, I would like to invite Dr. B. Stalin sir, Dean, Dr. MGR Education Research Institute of Campus Army, to felicitate the chief guest. Sir. Thank you, sir. Ladies and gentlemen, please welcome our distinguished speaker with a warm round of applause. Sir, the stage is yours.
So now we move to the second plenary introduction. It is our privilege to invite Dr. Ramchandran Sanwal sir, Assistant Professor, Department of Optometry and Vision Science, College of Applied Medical Sciences, King Saud University, Riyadh, Saudi Arabia. He will deliver a lecture on therapeutic efficacy of nano-formulated biomaterials in corneal wound healing and epithelial cell regeneration. To introduce our chief guest, may I invite Dr. Che Vadivelu, Associate Professor, Department of Biochemistry, to deliver the chief guest address.
Good morning to everyone. It is my great pleasure and privilege to introduce our distinguished resource person for today's session, Dr. Ramachandran Sanwal, Assistant Professor in the Department of Optometry and Vision Science, College of Applied Medical Sciences, King Saud University, Riyadh, Saudi Arabia. Dr. Ramachandran obtained his PhD in biochemistry from Annamala University and has an impressive academic foundation with an MPhil MSc in biochemistry and a bachelor degree from Madras University. He brings with him 18 years of research experience and 6 years of teaching experience at King Saud University. In addition to his academic contribution at Bangkok University Hospital, South Korea. His research interests encompass cell and molecular biology, corneal disease, nanotechnology, immunology, stem cell biology, biomedical imaging, and UV photoproduction. He also completed prestigious postdoctoral research at Tallinn University, USA, Tanvas, India, Bangkok University, and CH University, South Korea, making significant contributions to biomedical and translational research. Dr. Ramachandran has guided 14 postgraduate students. He published 25 peer-reviewed research journals in internationally reputed journals and served as a reviewer for leading scientific journals, including the International Journal of Molecular Science and Stem Cell Research Therapy. His research excellence was recognized with the Senior Research Fellowship from the Department of Science and Technology, Government of India. Today, Dr. Ramachandran will deliver a lecture on therapeutic efficacy of nano-formulated biomaterials in corneal wound healing and epithelial cell regeneration. His work integrates nanotechnology and regenerate medicine to develop innovative therapeutic strategies for ocular disease, offering valuable insight into cutting-edge biomedical research. Ladies and gentlemen, please join me in extending a warm welcome to Dr. Ramachandran Sanwal sir as we invite him to share his expertise and experiences with us. Thank you, sir.
Now, I would like to invite Dr. B. Stanan sir, Dean, Dr. Anjar Education Research Institute, to felicitate the chief guest.
Now I would like to invite Dr. Sheba Pesisman and Dr. Sumati ma'am to honor Dr. Sarveshwari ma'am, Head of Chemistry Department from Vellore Institute of Technology. Thank you all the dignitaries.
Good morning to all of you. I have to thank the management of this university as well as the other official members for organizing this 3-day conference, international conference on synergizing artificial intelligence and classical research in biosciences, engineering and natural science. So I have to thank the Dr. A.C. Shanmugam, the Chancellor of this university, and Trustee Madam Lalita Lakshmi for giving the permission to conduct all these events here, and similarly to A.C. Arun Kumar, President of this university. And then I have to thank joint registers here, both the joint registers, Peruvalidi and Saravanan sir for this campus, and the deans are Professor B. Stalin for this wonderful opportunity. And really I have to thank the conveners, R. Sumati Madam as well as the Selvi Madam for bringing this into the reality. So because I have convened 2-3 international conference, so I know that how much we have to work for that one to bring all the peoples here. So bringing the resource persons is one task, the another task is bringing the audience to the forum is also another task. So both is going to be a very tough, and we have to take care of all the sides, and so that in that aspect I have to thank both the conveners, Sumati Madam as well as the Selvi Madam, and similarly the cooperation from the department is also very much essential. So unless otherwise the cooperation is not there, we cannot do anything, and so that I have to thank all the department faculty members who are involved, directly and indirectly involved in this one. So the list is very long, so I can say that Sutagar, Fatima, Grace, Bhuwana, and then conference chairs, Siba, Felix and Balasubramaniam, so like that. So list is very long. So suppose if I didn't mention or fail to mention some names, please don't take it as a very serious, because I cannot go through all the names and I have only this much. So with this, thanks, I can go for today's talk, and I am going to give the lecture on modified Doppler hydrogen transfer and Suzuki-Miura cross-coupling reactions in the synthesis of quinoline moieties. Why I have taken the quinoline? Quinoline, actually this session is going to be on a hardcore organic chemist, but some applications aspects we can find it. Why I have taken this quinoline is actually anti-malarial, well-known anti-malarial, still it is used in the drug, and so that I have taken this one. And it's okay. Okay. Color contrast. Is it? Slide only. Okay. Okay. Na patala. So if you are checking this, how much amount of drugs are available worldwide, you can say that most of the drugs are nowadays with nitrogen heterocycles, and here you can see that bioactive heterocycles in bioactive molecules, you can say that simply aromatics are 10%, aliphatics are 4%, organometallics are 1%, but heterocycles, the bioactive molecules which are available in the, so far in the market or any research, nearly 85% is belongs to the nitrogen or generally you can say that a heterocyclic chemistry. So, cyclic, what do you mean by the heterocycles or cycles? It is going to be in a compound which is going to have the cyclic nature. In that cyclic nature, you are going to have heteroatoms. Heteroatom means apart from the carbon, what are all the other atoms are present? Generally, nitrogen, carbon, oxygen, it will be present. So, this kind of motifs only going to have the bioactivity, most of the compounds, and you can see that nearly 85% of the molecules contribute to this one, and so that which is having an interest. So, here are some of the examples, penicillin, acyclovir, like that, so many drugs which are available, and all are, you can see that which is having an either nitrogen or sulfur or oxygens are present, and so that these are all belongs to the heterocyclic chemistry only. Next one. So here I am very particular about
Use this laser, sir. This one. Yeah.
okay. So if we go through this one again, okay. So here you can see that number of heterocycles, here the nitrogen, oxygen and all those things. In this one, I am very particular about 6-member nitrogen heterocycles, and that nitrogen heterocycles only we are going to deal it here. So 6-member ring with a nitrogen, you are going to call it as a saturated ring is a pyridine, and unsaturated ring you are going to call it as a piperidine. So this pyridine ring is fused with another ring, then you are going to call that as a quinoline. So that's what we are going to discuss. Again, either it's a pyridine moiety or a piperidine moiety. You can see that number of drugs, whatever it is available here, you can see that omeprazole, isoniazid, nifedipine are like all these things are from the pyridine moiety, and piperidine also you have number of examples, and indole, pyrimidine, imidazole. So all these things are going to be in a classical. Next one. So in this case I am going to talk about only the nitrogen ring with a 6-member ring, and that is attached with a benzene ring, and this one we are going to call it as a quinoline. So this one, since these two rings are attached within 2 positions, and you are going to call that as a fused heterocycles. So I am very particular about that. That means our group is especially working on this fused heterocycles. So this fused heterocycles nearly back side. No, previous slide, previous. Yeah, it's okay. So you can say that nearly 60% of the new molecular entities contain at least one fused heterocyclic ring systems, and most of them are, you say that cancer drug activities, and the number is more than 75% it is available here. So as compared to the monocycles, fused heterocycles bind strongly with kinases, because whatever you are going to say the proteins or anything else which is going to have the nitrogen and oxygen, and it is going to have the lone pair of electrons, and here also you are going to have the nitrogen and hydrogen, and so that there is a hydrogen bonding interaction between the what you are going to have the peptides or proteins or whatever may be, and similarly here you are going to have the nitrogen and all those things, and so that this will have the very good interactions, and because of that only the heterocycles is playing in a very good role, or that is a vital role it is playing it, and that's why the points are here, hinge binding. That means perfectly it will bind with whatever the proteins you are going to take it or anything else, and selectivity in the drug, and then it is having a very good potency. All these because of the nitrogen or any other heterocycles present in that moiety, and that is binding with our selective proteins, and so that which is going to have that much activity. And then next slide please. So I am very particularly going for the quinoline, and again I don't want to take more time on this introduction itself. You can say that it is having a number of activities. With this moiety you can find antimalarials. It is a well-known quinoline drugs are antimalarial, antibacterial, antiviral, anti-cancer, antifungal, like that it is having. And apart from this medicinal activities, you can say that it is used in the dye productions, agrochemicals, as well as used as a solvent in the chemical process. Next slide please. So these are all some examples which are available in the market as a drug within this two. You say that this is the core of a quinoline, and you can see that all the cores are present in this drug, and this is chloroquine, quinidine, tafenoquine, all these things are quinoline based drugs which is available in the market. Please, next slide. So these are all the examples for, you can go ahead. Yeah. So these are all the examples for some other drugs with, you can say, quinoline moieties, anti-malarial, anti-cancer, anti-tubercular, anti-inflammatory. Next, please. So these are all the drugs with breast cancer, glycosidase inhibitors, antifungal, antibacterial, antioxidant, and it is having an antimicrobial as well as the DPPH scavenging activity, antioxidant property. Next one, please. So apart from this medicinal activities, which is having an dye activities also you can find out. So here all those things are there: antimicrobial, anti-cancer, quinoline dyes for bacterial application, and inhibitors, DNA, RNA imaging. So like that a number of molecules are. Next, please. And so up to this moment, up to this we have seen that what is the significance of quinoline, how much it is available in the market as a drug, and what is the other significance. Then we are going for the synthesis, how to synthesis, because in most of the cases the natural product itself which is not going to in the quantum wise which is not going to be satisfy all our requirements, so that we are forced to synthesis. So while you are going for a synthesis, how to synthesis, and for that here general synthesis methods we are dealing with nearly 6 methods. One is Combs quinoline synthesis, Cournot-Limpach synthesis, Dobner reactions, Dobner-Miller, Gould-Jacobs reaction, and Scroop synthesis. Here only we are going to do this reaction. I have modified, our own scholars they have modified, and what is the modification, that only now today we are going to discuss. Okay, so next one please. So apart from this synthesis, there is a one more synthesis which is called as a Freilander synthesis, and here also they are taking an aminobenzophenone which is going to condense with an alpha beta unsaturated ketone compounds with acetoacetic ester or acetoacetanilide, and then you are going to get an quinoline molecules. So this is also a well-known famous reactions. So all these are all some examples. I am going to deal only with this Dobner reactions. Please, next slide. So these are a literature survey. You can skip. Yeah. Then it is just an introduction. If you have an interest to know what is a multi-component reaction, actually while you are going for a synthesis, A you can convert it to B, B can be converted to C, or like that you can go on sequential synthesis. The drawback with the sequential synthesis, one step to the next step you will get 90, and another one B to C 90, and if you are going on like that, what will happen, the yield of the product will go on decrease. If you are going for a 6th step or 7th step, what the yield you are expecting, that much you will not get it. For that, if you are going with a multi-component reactions, so instead of having two reagents, now all the reagents are going to be mixed, and so that you are going to get multi-component reactions. In the multi-component reactions, you are going to say that all the required reagents, more than 2 reagents which is going to be combined, and you are going to get the yield. But here what is a major disadvantages, yield may be sometimes more, but you will get on a side products. Side products means what is your main product. Apart from that, unexpected products also you will get it, that you should know how to control. So if you know how to control, then this multi-component reactions is going to be in a very good reactions, and here what we are going to discuss is multi-component reactions. Please, this one you can skip. Yeah. So now I am going for one of the quinoline synthesis. Quinoline synthesis, what I want to have, want to synthesis is 4-quinoline carboxylic acid, and this 4-quinoline carboxylic acid, what are all the 2 reagents I can take it is, one is, this is called as isotene, and it is going to combine with an aldehyde, and I can prepare this 4-quinoline carboxylic acid. And this reaction, it is a naming reaction, it is called as a Fitzinger reactions. Another one is, you take the aniline. This is a very simple, wherever you go, any small lab, even with an +2 standard, all the chemicals, whatever. That's why I have taken this reaction today, because this is aniline. Aniline is available even in a 10th class or +2 lab. Okay, so aniline, and this one is any aldehyde, benzaldehyde, or any substituted benzaldehyde. And then this is pyruvic acid. Most of the biologist, you know that pyruvic acid where you are getting it. So this is pyruvic acid, very simple, and benzaldehyde 100 rupees or 200 rupees, 100 grams you can get it, and aniline also maybe. So with that is enough to start these reactions. Okay, so if you take these reactions and if these three are combined together, and you are going to get a 4-quinolyl carboxylic acid. So out of this, next slide please. Out of these two reactions, fixing a reaction we didn't prefer, but first we will see that what is the application and what is the drawback of this one. And using this reaction you can go for a wide range of product. Wide range of product means here you can go for a substitution, number of substitutions if you want to bring it with that kind of reactions you can do it with Fitzinger reactions. Another one is typically carried out with a large excess of alkali. Alkali means nothing but your sodium hydroxide or potassium hydroxide. You are supposed to use a large amount of sodium hydroxide or potassium hydroxide, and because of that what is happening, a strong base hydrolysis, the hydrolysis is going to happen in this position in the isotene, and then this is going to react and you are going to get it. So amino group of this intermediate condenses with the carbonyl compound to form the imine. This imine tautomerizes to the enamine, and which undergoes the cyclo dehydration, and you are going to get this product. What is the drawback of this reaction? Self-condensation of carbonyl compound. That means self-condensation means you have already studied in our plus 2 or 10th aldol condensation are like that. One aldehyde will react with another aldehyde. So if you have, because while you are having a ketone, one ketone will react with a same molecule of another ketone, and so that the condensation will be more, and that's what we are saying, that it is a multi-component reaction. In that one the self-condensation, otherwise you say that byproducts will be there unexpected. You are expecting this one, but apart from this product you will get something else. Something else, why? This is going to react with another molecule in presence of a base. It is very well known reaction in our aldol chemistry, aldol condensation reactions and all those things, and so that such kind of reactions will happen. So self-condensation of carbonyl compound, especially with the aldehyde, it is going to be there, and due to the steric hindrance, alkyl aryl ketones with the structure of this one does not undergo the reaction, and major thing is strong basic conditions degrade the sensitive substances. That means even though you are saying that variety of products are possible, while you are involving an alkali, you cannot get, you say that some of the reactions are going to be, reagents are going to be sensitive, reactants are going to be sensitive, and so that you will not get the expected product, and so that this is the drawback of this one. Next slide please. So next one is our own Dobner reactions. In this Dobner reactions, it is versatile, efficient method to construct the quinoline 4-carboxylic acid. It is also a powerful tool in the diversity-oriented synthesis. It is used as a wide variety of substrates, and a recent use of Dobner reaction is going to be used as a quinoline-linked COFs. Okay, carbon-based organic frameworks. You can do that. Next slide. And in this one, so far in literature, what are all the conditions are available? Conditions are here. Using these three reagents, you can go for an amino sulfonic acid with the water and reflux. You can get this quinoline carboxylic acid. Otherwise, the same reagent, use the sulfuric acid. Otherwise, you can go for ytterbium PFO3 with the water reflux. Otherwise, a BF3THF. And all these things, one or other way, is not good. Okay, so here you are going to use excess amount of sulfuric acid, amino sulfonic acid, and the conditions are going to be very strong conditions you have to involve. And next slide please. So here what we have modified is, we have used para-toluene sulfonic acid. Instead of using a sulfuric acid and other things, we are using an ethylene glycol. So in the green chemistry, ethylene glycol is a very good solvent, and which is not going to be that much effective in environmental pollution, and so that we have used the ethylene glycol, and which is going to be with, backslide. So here we have used the ethylene glycol and water, which is going to be in a 2:1 ratio, and the temperature, you see that in the last slide I have shown that only a refluxing condition. Refluxing condition means high temperature, nearly 78 or 100 or like that. What is the solvent you have used? Accordingly, it is going to be in the boiling condition of that solvent. So high temperature is involved. But here we have used only the 50 degrees Celsius, and reaction is going to be only 3 hours. In this conditions, we have modified this reaction. So reaction is a one-part simple synthesis, and then very faster, reaction is going to be within 3 hours, not for 24 hours or 12 hours, and so that this is very faster and greener approach. Atom economy. What do you mean by the atom economy? Means in a molecule how many atoms are there and how many atoms are converted to the product. So that ratio you have to convert, and it is a rule of green chemistry also. So here you can see that atom efficiency is going to be very high in this one compared to the earlier case, and then low toxicity. Whatever the chemicals here it is involved is less toxic, and so that this approach is going to be a very good one. And how we have used this, in the earlier slide if you have seen that they have used only the pyruvic acid, but what we have used is sodium pyruvate. So while you are using a sodium pyruvate, the solubility is going to be very good, and because of that the reactions are happening in a low temperature itself. And so here you see that sodium pyruvate in the place of pyruvic acid, superior solubility and ionization properties, that is going to promote the efficient hydrogen transfer under the intermediate stabilization. And the use of parasulfonic acid as a branched acid, it is enabling the multiple functions including the skip base formations. The skip base formation, next slide I will show you that what is a skip base formation and all those things. Then followed by the hydrogen abstraction, cyclization, and relatively mild thermal condition without need of toxic or corrosive reagents. So this is the important modifications we have done. And then this one we have patented, as well as next slide please. We have published also. And here what is a skip basis? Skip basis, though we are saying that a skip base is a scientist name, any aldehyde going to react with an amine function, so just a water molecule condensation, you are going to get a C double bond N. That functional group, if you are having it, you are going to call that as a skip base. But here the mechanism also we have reversed, because in the earlier mechanism what they have so far produced in the literature, they thought that it is through the skip base condensation, but here we have trapped the intermediate, and this trapped intermediate which is going to have the C, what is that? This molecule we have find it out. That means this aldehyde condenses with an amine, and you are going to get this compound, and this is going to react with an pyruvic acid. So actually the mechanisms are already in other way. You say that these two are going to react and then finally this one, but we have reversed, and then this intermediate we have collected. And this intermediate, so far intermediates are very much essential. Why? Means if you want to give the mechanism, and intermediates are necessary. So intermediates also we have trapped in these reactions, and this intermediate, if anyone knows the spectrum, suppose you can see that we have CH2 group is here. I don't know whether any organic chemists are here, and you can see that this is a carbon hydrogen co-C, and that these two hydrogens are placed here, and that is attached with a single carbon, and that is an evidence for CH2 is present in the intermediate, and because of that this mechanism we have proposed, and the intermediate also we have isolated, and we have revised the mechanism of this product is also. Okay, so next slide. So then after first you say that you have to stabilize the reactions. So we have gone for all kind of optimizations, and optimization has been done. Then we have to apply whether only one molecule you will get it, otherwise if you are going for a substitutions, number of molecules you can produce. Yes, we have gone for such kind of things. We have produced nearly, you see that, so 29 compounds we have prepared. So all are within various substitutions. Actually in this Dobler reaction so far they have produced only 7 or 8 derivatives only. But here we have produced 29 or 30 derivatives, and that is a remarkable achievement in these reactions. Okay, and next one. So it is, while modifying it, if you are having a halogen in substitution, that means in your compound if you have the halogen, that is also going to be withstand, and then any electron withdrawing or electron donating groups are present, that is also working, and not only the pyruvic acid, and you can go for any other COOH groups are available. That kind of reactions also very much you can do that. And this pyruvic acid is actually 10 gram is 2,200 rupees. But since we have involved a pyruvate, sodium pyruvate, the cost of that one is, you see, this is 10 gram is 2,200, but this 25 gram is only 500. So the cost of production is also brought down, and so that industrially this is having a significance, because industry is mainly looking for cost cut. how to reduce the cost, and so that here if they involve this one, and you can do that. Okay, so with this, next slide. So after that, the amine what we have produced, that is converted to the amide, and we have produced nearly 10 or 15 amides only we have produced. Why means, and with this 29 carboxylic acid, quinoline carboxylic acid, if you are going for a number of amides, maybe 100 derivatives you will get it. But all the 100 derivatives we didn't prepare. What our scholar did is, and he has gone for docking studies. In the docking studies, which is having a very good activity, and those molecules only we have produced, and so that only 29 compounds here it is produced. Next slide. And the mechanism, next one. So here also you can see that, yeah. After that, this is why I have shown here is, you can use this Schrödinger software, and you can find out that what do you mean by the HOMO, highest occupied molecular orbital, and what do you mean by LOMO, lowest unoccupied molecular orbital. That means where the electrons are there and where the unoccupied orbitals are there. And these two energy levels you can find out. If the energy gap is more, the stability of the molecules is going to be more. If the gap between HOMO and LOMO is going to be very less, reactivity will be very high. And to find out that one, theoretically, it is not necessary to do it in the practical. So such kind of softwares are available. Even you people also can involve in that one. That's why I have shown this one. So using the HOMO, LOMO, using that software, you can find out what is the energy gap, and theoretically you can find out of this many molecules you have synthesized, which one is going to have the more activity, which one is going to have the less activity. Such kind of things you can do that. Next slide. So that is what it is given here. The energy gap, what it is given is negative of 4.169, here it is 2.782. That means this is going to have the more activity compared to this one. Like that, it is theoretical prediction only, softwares are available. Okay, so that one, you, suppose the institution is not in a position to spend more money at this initial stages, you can go and download this kind of softwares which is freely available, and that kind of things you can do it. Okay, next slide. So and this is going to be in a electrode, what is that, mapping of electrode potential. In this one, what you can see this, again it is used in a theoretical software only. In this theoretical software, where the red color is more, the electron density in that part is going to be more. Where the blue color is going to be there, electron density is going to be less. That means this is going to have the negative character, and this is going to have the positive character. So most of the time the reactions are going to be with a positive, negative, negative, positive, like that. So theoretically you can predict. Okay, so next one. So these are all for all the compounds, they have find it out. What is a HOMO, what is a LUMO. Only 2-3 compounds I have shown it, but using this software you can find out for entire molecules. How many molecules you have synthesized, with just on a software you can do that. Okay, next one. So then they are going for docking studies. I think for medical people or paramedical, I am not a person to explain, because you are the people who have to explain to me. So they have selected 2 or 3 proteins from the PDB, and that is going to be docked for the further activities, and then we find out that what are all the activities here. So this one I think no need to explain. You people are master than me. Okay, so next. Okay, so like this we have find out the energy gap for another 30 compounds. Thank you. Next one. So again it is going for a binding with the docking with the protein what they have selected. It is giving a very good activity. Next one slide. Yeah, next one. Okay, so okay, thank you. Thank you. Any other interactions or questions, or if you want?
Actually not a query, it's a request actually. If in case, can we use your research assistance if we are working in docking, because we are actually looking for molecular docking.
Actually it's a research assistant which you are looking for. If in case they are in need, can we avail your research assistant for molecular So,
We can do.
Yes.
Our own scholars are here also that there are, now itself we can go on and interact and if you
Yes.
want to give them, they will.
actually, we
Yes,
have
sir, I'll
seen-
share the
yes,
That kind of helps you. That's why we want to introduce the things here.
yes.
So how we do that, that can be done. So
Thank you, sir. Thank
just you need an guidance, how to download, how to use that one. That is,
Yes, sir, I know the basics of bioinfo. The thing is, like, I need little assistance in working on this protein ligand interaction.
yeah, that scholar is here. What he has done, Sachin, he is sitting in the last. Okay, so that can be done. Otherwise, just write to me, so I will direct the students. No, it's not issue. Thank you.
Very nice presentation, sir. And my project also the same, but I have concluded with the thiazole. And one more question, I have done in the microwave. What is the difference between the microwave and one-part component, sir? Whether which will be preferable?
The reaction can be done in thermal method. In the thermal method means you will mix all the reagents and put it in a solvent and heating is you are going to give it. Instead of a heating, thermal conventional heating, the heating you are going to use the microwaves. That's all. So that you are going to call that as a microwave assisted reaction. No, result will be different because what the content, heat content you are going to give it in a microwave, that will be very large. So reaction will be very fast and what is the power you are going to select it, and based on that sometimes course of the reaction will be changed, product also will be changed. Thermal reactions generally it may take 12 hours or 24 hours, but microwave reactions sometimes even 5 minutes. The very first reaction madam did the Friedlander reactions, and if you are going for that conventional reaction it may be 12 hours to 24 hours. But what she did is that same microwave reaction 5 to 10 minutes. That's all. That depends on who you are going to select. And apart from this, one more is also there, sonochemistry, sound waves. So instead of a microwaves, you can use the sound wave. Sound waves also you can produce the reactions.
The reaction will be more faster than that.
Yeah, more fast.
Sir, and how is the response from the industries, as you said, 2,200 to 500 rupees?
So generally, if you are speaking it, this we are publishing it.
Okay,
They will go through, no need to contact us.
okay.
They can copy. That is one, because that is, majority of the times we are dealing with academic research. We are not bothering about who is going to use it. And another side, if you are checking the industry, they might have powerful reaction than this one also, but they won't reveal. Because if they reveal, someone will steal that, and they are all competitor only. So if you are running an industry, I am running an industry, then I will see you as a competitor, you will see me as a competitor. So we won't reveal what is the reaction conditions, what kind of setup we are having it, that we won't reveal. So that is the case. So either they have it, they don't have it, I don't know. But even if they don't have it also, it is already published. They will take it, that's all. No need to get my permission.
Thank you.
Thank you very much, Professor Vijay Kumar sir, for your insightful and thought-provoking lecture. Your valuable research findings have greatly enriched our understanding of modern synthetic chemistries. We sincerely appreciate your time and expertise, sir. And I will move on to the final plenary lecture of Elevate 2026. Now I invite Dr. Ramachandran Samuel, sir, Assistant Professor in Department of Optometry and Vision Science. The session is yours, sir.
First of all, good morning to all. I would like to present my research experience. Before that, thanks all the coordinator and organizing members. Today I am going to talk about my research experience entitled Nanoformulator Biomaterials Anti Corneal Wound Healing and Epithelial Regeneration. Today my topic is all over covering corneal regeneration and wound healing, because I am an ophthalmologist, so that's why my study is focusing on that area. So sorry about that, other area chemist people. Maybe I hope to cover all the area. Anyway, next slide. This is the outline of my talk, especially corneal structure and functions and its degenerative disease, pathogenesis, and as well as today I am going to talk about elaborately with BSA formulator hydrogels. Nowadays that contact lens and some other hydrogel based products, and especially it's used for biomaterials. So today I am going to talk about BSA formulator hydrogels, especially its therapeutic efficacy and future perspectives. Next slide. Yeah, this is the cross section of whole eyeball. They have two chambers, anterior chamber and posterior chamber. Anterior chambers is covered with cornea. This is the cornea ring. Okay, that posterior chambers is covered with iris, lens, vitreous body, and this is the retinal pigment. That retinal pigment has 7 layers. This is the outer scleral layer. This is the retinal pigment. Center have corticoid pigments. That edge, this is the optic disc. That blood vessels and nerve fibers is connected to center of the optic disc. That optic disc is connected to optic nerve. This is the optic nerve is connected to our brain, center brain. This is the function of eye. So when we look the anterior chamber, this is the cross section of cornea. They have cornea, they have 5 layers. Top, this is the epithelial layer. Before that, they have T-films. The T-films only cover lubrications of our eyes and it protect foreign bodies and other pathogenesis. Next, this is the epithelial cells. Epithelial layer, they have 3 kind of epithelial cells. That surface have superficial epithelial cells. Here it's come superficial epithelial cells. Center one is wing epithelial cells. Then third one, this is the basal epithelial cells. That basal epithelial cells is connected with Bowman's layer. That Bowman's layer is connected with stroma. That is stroma made up of collagen fibers and keratocytes and fibroblasts. This is the main component. It's connected with epithelium and desmus membrane and endothelium. This is the 5 layers. It's present in surface, top of the eye. This is the anterior chamber. I am going to talk about my study. Whole study is covered with anterior chambers, that corneal degeneration and corneal wound based disease models. So according to that, This is the cornea. Cornea is transparent avascular tissues that contribute approximately 2/3 of the eye refractive powers, especially that corneal epithelial integrity is the critical for maintaining optical clarity and protecting underlying stromal tissues from microbial invasion and environmental damages and foreign substance. As well as that corneal injury caused epithelial damage, stromal remodeling, and tissue fibrosis and vision impairment, such as vision loss. Next slide. Yeah, this is the list of corneal injuries based degenerative disease, especially stroma. This is stroma. When we got some injury or some machine made or some accident basis that cause corneal damages. Yeah, this is the chemical bonds, especially whatever we used chemicals or some substance or some salt, alkali content, it causes like that. That cornea, total cornea and epithelial layer went out and it caused white color shadows. Yeah, this is the infections, especially viral infections, bacterial infections, and some other microbial content. It caused like that. That is the total. This is the conjunctiva. First of all, that conjunctival epithelium as well as blood vessels is migrate into cornea, center of cornea. That cause vision loss. Yeah, this is the post-surgery failures. Nowadays we are suffering cataract and so many keratoconus and so many diseases. These all the disease, the end solution is we have to do transplantation. There is no powerful treatment. So that for that transplantation, that sometime when we transplant a new cornea, it won't adjust to our body because it is heterogeneity. Because of that heterogeneity, it's caused failure, the transplantation failure. That conditions, we got vision loss and the total vision goes disappear. So for that, this is the one of the category. And next one, this is the degenerative disease. Nowadays, we have so many incidents happen in the degenerative disease, especially pterygium. Pterygium, our area, we have lot of outside bodies and dust and even sun rays. Sun rays, that ozone layer, it's penetrating into environment. It directly comes to our eyes. So even though we are in hot places, so that sun heat also may cause that pterygium. When we have suffered that pterygium, that conjunctiva tissues is migrate into inside the cornea. It's covered like bubbles like that. So for that it's come, this is degenerative disease. So yeah, these all are the list of corneal injuries. When they happen like that, this is the cross section of that cornea, that damaged cornea. You see, first earlier slide I showed you that structure of cornea, that surface have epithelium, center have stroma, and bottom have desmoid membrane and endothelium. For here, you see, this is the epithelium. Between the epithelium, that tissues, that fibrous tissues are penetrated. This is called scarring, corneal scarring. When we happen this, that whole epithelium and stroma, it will degenerate. It won't come to normal conditions. This is the conditions of corneal injury as well as degenerative disease. Next slide. Freeze. Yeah. Can you adjust that? This. Yeah, this slide shows list of corneal inflammatory response as well as tissue fibrosis. Yeah, when the cornea or epithelium is going to repair abnormal conditions, first of all, the list of cytokines and growth factor activated. Those activated cytokines and growth factors and growth factors, it's activate keratocytes and as well as fibroblast. Those keratocytes converted to, differentiated to fibroblast. That fibroblast is differentiated to myofibroblast. Those myofibroblast, it activated TGF signaling. This TGF beta is the main key factor for causing tissue fibrosis. Yeah. Yeah, these are the growth factors and cytokines. It's mainly activated chronic inflammation. That chronic inflammation condition increase TGF beta levels. Those TGF beta, it differentiated to myofibroblast to myofibroblast. Yeah, this myofibroblast leads to cause corneal fibrosis. Next slide. Yes. Yeah, this is the this slide shows corneal wound healing, especially corneal wound healing when we use alkali, especially sodium hydroxide or some alkali substance, is mainly cause stroma, that collagen degenerations. These are the collagen fibers. When we applied that alkali solution denatured to collagen fibers as well as inside the cells. That condition, TGF beta will activate it. Yeah. As well as same time, when we applied the alkali or some substance, it disturbs tear films, especially surface that tear fluid levels. It goes to dry eye conditions. Yeah, next slide. Yeah. This is the current treatment option as well as limitation. They have some certain limitations. That current treatment options are artificial teeth as well as anti-inflammatory drugs, antibiotics, amniotic membrane transplantations, as well as corneal transplantations. For that treatment conditions, they have some limitations, especially limited degenerations, donor shortage, recurrence, and tissue fibrosis. As well as nowadays we are very famous for three-dimensional hydrophilic network mimicking native extracellular matrix, because that is made up of extracellular matrix proteins as well as matrix substance. So when we disturb that extracellular matrix, it may disturb all the functions. Next slide. Yeah, this is the this slide shows list of biomaterials as for used corneal regenerations. These are the biomaterials, list of biomaterials, especially collagen-based biomaterials and decellularized extracellular matrix cornea. Nowadays, not nowadays, earlier, it's very famous technique. We collected to outside the cornea room and we removed all the cells, keratocytes, fibroblasts, and whatever have nucleated cells, we have to remove out, and then we have to make a matrix, that powder shape, especially that collagens and elastin fibril fibers. We make matrix. That matrix then we will reshape with contact lens like that, contact lens like material. So that one very famous earlier, but they have some limitations as well as that gelatin based biomaterials and silk fibrin and polysaccharide, especially alginate and chitosan, hyaluronic acid, cellulose. And as well as this is the synthetic polymers, especially PVE, PCl, PLGA. These are the advantages, and even though they have some limitations, especially when we use collagen-based biomaterial, it induces immune responses, particularly when originating from various species. When we use homogenesis, that same pieces, they won't bother anything. When we use that foreign bodies, that heterogeneity, it won't adapt easily. It may cause some immune responses, autoimmune responses. Yeah, as well as enzyme degradations, physical factors, and chemical agents all possesses risk factors. Yeah, as well as the production can be expensive, particularly when it involves large quantities and specific properties. Yeah, when we use that extracellular cornea, it may require the removal of residues, antigens, after decellularization and remain the cytotoxic agents used for decellularization protocols. Also, when we used gelatin-based biomaterial, it needs excessive biodegradability and inadequate mechanical properties. And as well as when we use silk fibrin material, for that we need standardizing raw material, and their processing methods is challenging, particularly difficult to stabilize for long-term perspectives. When we use polysaccharides and those material, that one also, the final shapes and quality of corneal implant may be fluctuate due to batch-to-batch discrepancies. Especially when we use chitosan-based material as to potential to be recognized as the foreign immunogens. Yeah, that last one, that is synthetic polymers, also have some limitations, especially demands comprehensive processing and chemical modification. PCl has poor hydrophobicity and transparency. Next slide. Because of these limitations, we need to find out new biomaterials, especially bovine serum albumin or human serum albumin biomaterial. Because bovine serum albumin, human serum albumin, those substance we always used for cell culture. If you anyone know about cell culture in vitro study, that BSA is the main core substance. It activates cell proliferation, migration, and invasions. Because of that purpose, we have chosen BSA as well as human serum albumin. For this BSA have high biocompatible, biodegradable, non-toxic, abundant, transparent, and easy functionalization. We will modify the models. Whatever we need that mold, we will prepare easily, and it is flexible and as well as very high transparency. So for that, even though when we use BSA or HSA, it's very easy to cross-linking with other substance, and even functional group binding also very easy, and incorporation, that is functional group incorporation, so bioactive molecules. Another one, easily we will control the drug release. The next, it has have high water intention, oxygen permeability, and transparency, mechanical stability. This BSA-based biomaterial, we will combine with drug molecules as well as drug delivery of riboflavin, retinoic acid, ascorbic acid, growth factor, antioxidants, and other natural compounds, biomedical compounds. We will combine each together, and it may activate more biological process, as well as it promotes cell migration, proliferation, and re-epithelialization. Next slide. Yeah, This is the our study experimental models. Here we developed ex vivo as well as in vivo model. First, the main concept is to evaluations of bovine serum albumin formulated hydrogels containing riboflavin, retinoic acid, ascorbic acid for corneal wound healing and epithelial regenerations. Yeah, this is the ex vivo organ culture model. For that we use 6-well plate. That 6-well plate, first we made agarose mold. This is agarose mold. This is the 6-well plate. After that, this is the corneoscleral rings. Corneoscleral rings, we have to cover the dome shape culture. This is the medium. This is the transparent airway models. Okay, these are the camel cornea. We cultivated, incubated with 6-well plates. Okay, yeah. Next one, this is the in vivo alkali bone in rat model. Yeah, here we use 3 mm paper disc. That 3 mm paper disc we soak with sodium hydroxide solution. That sodium hydroxide solution we have to implant surface of the eyes. Okay. Yeah, next slides. Yeah, this is the BSA based hydrogel preparation composition. For BSA based hydrogel composition, we used 10% BSA. Here we used different grade BSA. Finally, we optimized 10% BSA, riboflavin, retinoic acid, and 2.5% glutaraldehyde as a cross-linking agent. Here we used very less concentration because this glutaraldehyde causes some toxic materials, so that's why we want to reduce this concentration. Instead of this glutaraldehyde, we use that UV cross-linking. Here we followed that 10% BSA as well as UV cross-linking. Here this is the UV cross-linking process. After UV cross-linking, we used that membrane transplantation, that membrane dialysis. This is the membrane dialysis. After membrane dialysis, we prepared, next slide. We mixed with growth medium. When we use that growth medium, yeah, first we used the ex vivo organ culture. Here we chose the camel cornea. That camel cornea we where we wounded 8 mm diameter, 250 micrometer depth. Those cornea we used cultured for 24 hours, 48 and 72 hours with and without BH8 compound, that hydrogels. Okay, yeah. Before incubation, first we created corneal wound. Once created that corneal wound, we peeled out that epithelium. Okay, once that peeled out that central corneal epithelium, then we cut it that corneoscleral ring. Once we take out that corneoscleral ring, then we used that culture, that ex vivo culture. This is the ex vivo culture. We used 3 groups: control and without treatment and with treatment with corneal wound. Then this is the in vivo study. Here we used alkali rat bone rat models. Here we used 1 molar sodium hydroxide in 3 mm paper disc. Okay, this is the anesthetized rat. Once anesthetized rat, then we used 3 groups. This is the control rat. This is the alkali bone rat after 7 days. Okay, yeah, this is the alkali plus BHA hydrogel treated rats after 7 days. Okay, after 7 days, next slide. These experiment ex vivo and in vivo studies, after 7 days, we After 3 days for X-ray, we take out the tissues and we did tissue processing for biochemical analysis. As well as in our study, after 7 days, we sacrifice those mice, then we collect out that whole high bulb, then we use it for biochemical analysis, that tissue process. Okay, yeah, this is the ex vivo results, for ex vivo organ culture results. These are the different time point that cross section of cornea. Yeah, this is 0 hours cornea, this is 24 hours cornea, this is 48, and this is 72 hours. When we see that control cornea, they have clear epithelium, stroma, and desmus membrane. Okay, that 0 hours we peeled out that epithelium, that center of cornea. With here, this is the without cornea epithelium showing stroma and desmus membrane. Okay, yeah, this is the 24 hours cornea. Without hydrogel treatment, that epithelial migration, this is the epithelial migration, is very less compared to BHG treatment. Okay, yeah, that both sides that epithelium come to near center cornea. Okay, yeah, when we see that is 48 hours, yeah, this is the without BHG treatment. Here you see this is the made some scarring, that epithelium penetrate inside the stroma. Okay, yeah, when we treated with the BHC, that epithelium linearly migrate surface of stroma. That stroma also is very clear compared to that untreated groups. Okay, similar 48 hours that untreated groups is make that scars. Okay, this is the epithelium make scar, but BHG treatment, that epithelium almost similar to control. Okay, yeah, next slide. Yeah. This is the Nile and Dapis staining confocal images. When we use that confocal images, we will easily differentiate to epithelium regeneration as well as stromal regeneration. Okay, yeah, this is the control group. When we see that 0 hour control group, yeah, this is the epithelium, this is the T film. Okay, this is stroma. Okay, this is the Bowman's layer. Okay, that intermediate layer. Okay, when we peeled out that epithelium, this is lack of epithelium. They have only stroma. Okay, yeah. After 24 hours, without treatment, the epithelium, that basal cells is it won't differentiate well. But when we use BHG treatment, you see that the epithelium is differentiated to basal epithelium. Okay, that this is the stroma and this is the epithelium. Okay, yeah. After 48 hours and 72 hours, that BHG treatment is clearly differentiated to different type of that epithelial cells, that basal epithelium, wing cells. Okay, yeah, this is the clear structure of regenerated epithelium. Okay, yeah, next slides. Yeah, this is the electron microscope ultrastructure structure of corneal rearrangement. When we used electron microscope, this is the control, the A and B, this is the control normal control cornea. Yeah, this is the microvillus surface of epithelium. This is the epithelial cells, different types of epithelium. Yeah, this slide shows this is the Bowman's membrane. This Bowman's membrane present in normally. These are the basal epithelium, and these are the anterior stroma. That anterior stroma have that collagen fibrils and elastin fibers. Okay, these are the 72 hours wounded cornea. When we cause wounding, that basal cells is not differentiated as well as migrated. Okay, even that Bowman's layer also it's not well developed. Okay, yeah. Yeah, same, yeah, this is the Bowman's membrane degenerations, and as well as here the collagen fibrils as well as that epithelium also degenerated. Okay, these are the collagen bundles. Okay, this is the collagen fibrils. That collagen fibrils and bundles also is partially developed. Okay, yeah, this is the 72 hours with BHG treatment, treated cornea. You see, after BHG treated at 72 hours, they have different layers of epithelium. Yeah, this is the superficial epithelium, this is the wing cell epithelium, this is the basal epithelium. Okay, as well as here you see that the microvilli is also well developed, as well as that Bowman's layer also developed. Okay, yeah, next slide. Yeah, this is the between epithelium and stroma. Here, this is the well-developed Bowman's layer, as well as the collagen fibrils and collagen bundles also well-developed in BH3 treatment. Next slide. Yeah, this is the extracellular matrix regeneration, especially when we treated with the BHCC, it significantly increased fibronectin. Fibronectin is the growth factors, that surface growth factor is indicating that a brown color dye accumulations. Okay, yeah, this is the control cornea. When we treated with BHC at 48 and 72 hours, that accumulations of that fibronectin expression is very high. Okay, yeah, this especially in superficial epithelial cell as well as and basal epithelial cells. Okay, next slide. Yeah, this is another growth key factor Ki67, that stem cell marker. That stem cell marker also significantly increased in 72 hours, 48 hours BHG treatment, because this marker indicating different regenerations of epithelial cells, especially that they have different kind of cells. First, when we compare to limbus have limbus stem cells. That limbus stem cells, it's migrated to epithelium. That limbus stem cells is differentiated to epithelial cells, especially basal epithelium. That basal epithelium then differentiated to wing epithelial cells. That wing epithelial cells is differentiated to superficial epithelial cells. That superficial epithelial cells have microvillus as well as tape films. That lifetime is only 7 days. That once that superficial epithelial cells gone out, then again that cycle process will start. That limbus cell differentiated to basal epithelium differentiated to superficial epithelium. Wing epithelial cells differentiated to superficial epithelium. This is the cyclic process. When we are healthy and have good energy sources, this process will continue simultaneously. When we suffered some dry eyes or some disease or some environmental insult, this cycle will stop. When they stop this cycle, that epithelium it won't regenerate. That condition, that epithelium will cause degeneration and cause death. When they cause dead cell, that dead cell is stopped to productions of tooth fluid as well as tooth fluids. That conditions we suffered dry eye. Dry eye is the main sources for all other diseases, especially cataract and more severe disease. The dry eye is the main causes. Okay. Next slide. Yeah, this is the X-ray animal model. Yeah, this I already told you that this is the tooth fluid. When we collected from control rat, you see this is the surface ring, that tooth fluid ring. after dried that tear fluid, it's come ring. This is the surface ring. This is the core center ring. Okay, yeah. When we use that alkali bound rat, you see here they have infiltrated cells, especially so many that infiltrated blood all are combined and it stop the rings, that surface rings. Okay, yeah. This is the alkali bone plus BHA treated rats. You see here his ring. Depth is very high and even center core is very clear and there is no infiltrated cells. Okay, yeah. As well as this is the cross section of cornea, that anterior segment. This is the normal cornea. You see here, this is the corneal ring. This is the intermediate phase. This is the iris. Okay, this is iris and this is the lens. Okay. When we treated with alkali solutions, that cornea totally meshed, and even that intermediate phase also is collapsed, and as well as that center, that vitreous fluid, as well as that retinal pigment that cells are mixed, and it can infiltrations and fluid level degenerations. Okay, yeah. When we treated with the BHG treated after 7 days, yeah, you see this is the epithelial regeneration, this is the stroma, and this is the iris, is regenerations of iris. Okay, yeah. This is the posterior segment, retina. That retina, I already told you, they have 7 layers. Yeah, this is the clear layers. Sclera to that neuronal fibers. Okay, this is the neuronal fibers. This is the retinal pigment. This is the epithelial bodies. Okay, yeah. When we treated with alkali solutions, that whole structure will collapse, and even vitreous fluid also collapse. That this epithelial course structure is mixed with the whole globe. Okay, after treated with BHC, that layer come to back. Okay, that regenerations of that 7 layers. Okay, next slide. Yeah, this is the mechanism of that BHC, especially retinoic acid, riboflavin, BSA formulated hydrogels. This is the cross section of hydrogels. These hydrogels, when we applied with that wounded cornea, it activated fibroblast transplantation, transdifferentiation, especially promote Ki-67 mediated epithelial migration and proliferations, as well as that RA regulate Wnt and beta catenin signaling and stromal regenerations. Okay, next slide. For this, we have some challenges as well as future prospectus, especially BSA, riboflavin and retinoic acid hydrogel is the promising regenerative biomaterial for corneal wound healing and epithelial cell regenerations. They have some other smart hydrogels, especially we need to develop stem cell delivery, gene therapy, integration and personalized medicine for future prospectus, and it's need to animal elaborate studies as well as human trials and safety and efficacy of validations. That's next. Yeah, thank you so much for your attention. If you have any questions, please ask.
Sir, if the main goal is to mimic the extracellular matrix, means instead of hydrogel, have you tried any other fabrication techniques?
No, this is the basic study. For future, we have to going to migrate translation studies. Okay. For that translation study, we have to cover all the area.
Have you tried with only the cross-linking only with the glutaraldehyde, or else you tried various cross-linkers like
Yeah, first of all, we applied glutaraldehyde cross-linking. For that, some toxicity will happen,
Yes.
cytotoxicity. For that, we removed that glutaraldehyde and we use for UV cross-linking, UVA irradiation cross-linking.
Yes.
For that, we need a lot of time compared to cross-linking. Cross-linking we will use within few seconds or few minutes. Yeah. For that UV cross-linking, we need 24 hours like that. For time being, we succeed for UV radiation cross-linking, but in future we have to compare both of them, and we have to go for product development. Yes.
Is the hydrogel only applicable for optometry, or else we can use that hydrogel
Yeah,
for
we will use
other tissue
different-
engineering
yeah.
process?
Yeah, we
only
will use a
process?
lot of other studies also, especially center that nerves degeneration, as well as that stem cell transplantation, and even that tissue graft.
Yes, sir.
Yeah.
Because in recent years, the burn wounds are, there is no alternative solutions other than the grafts. So we can use for these hydrogels in the burn wounds also.
Yeah, that's why we are trying to develop for product. Yes.
Yes. Okay, sir. Thank you, sir.
Yeah.
Hello, sir. Very
Yes.
nice presentation,
Welcome.
sir.
Thank you.
Actually, I have a curious question. Many literature I have seen, people are working in the hydrogels, and is there any alternative options are there, like nanofibrous systems are there?
Yes.
Is it applicable for this applications?
Yes, yes. But according to our product, how
Okay.
we are making the product, that hydrogels, they have different grades.
Yeah.
That rough hydrogels have, that soft hydrogels have.
Yes.
For optometry, we always use soft hydrogels.
Okay.
Not rough. For biometric purpose,
Okay.
biological and some other purpose, for matrix preparations.
Yes,
That
bone tissue
bone
engineering,
preparation, that we use for hard hydrogels,
Yeah.
not
Okay.
soft hydrogels. That
Okay.
are, yes.
Okay, sir. And one more question.
Depending upon the concentrations.
Okay.
Yeah.
You have added 2% glutaraldehyde, right?
Yeah, 2.5%.
Have you tried with genipin? It quite non-toxic, right? Compare with glutaraldehyde, genipin is
Yeah,
little bit-
previously I used a BSA-based gold nanoparticle.
Okay.
For that, they used 25%.
Okay.
Yeah, 25% for other studies, not
Okay.
that optometry purpose or ophthalmology aspect.
Okay.
So according that formula, we are reduced that concentration
Okay.
25% to 2.5%.
Okay.
Okay,
It is too
then
less,
used
I
for cell culture, that is cytotoxicity, as well as that drug delivery aspect.
Okay.
Compare that 25%, that 2.5%.
It won't create any toxicity,
Even
right?
though after cross-linking, we used that membrane dialysis.
Okay.
That excess of unreacted glutaraldehyde,
Will be
it will
removed.
go out from the dialysis membrane. After that only we use for cell culture and cytotoxicity study. There is no excess of glutaraldehyde. That's why
Okay.
we used minimum concentration. But in future we developed that UV cross-linking instead of
Photo cross-linking.
Yeah, photo cross-linking.
Okay, sir. Thank you, sir.
Okay.
So thank you, sir. Like actually your work is very impressive. Like I have two questions, sir. First one is like you spoke about the graft rejections.
Yes.
Like there is a quite possible chances of the graft getting rejected, that the first few disadvantages before designing your bovine serum albumin based hydrogel.
Yes.
So if that is the case, you say you were like showing us the experiment which had this camel eye I would like to know how it will mimic the human thing, or will it have any replacement of this corneal transplantations which are in much need now?
Yeah, we will do. We have a lot of experiment. For presentation only, I displayed for camel eyes. We used even bovine eye also, and as well as human eye for XYO model.
But in general, for eye transplantations, will the graft rejections really work, or because it is actually anybody can like donate their eyes and the cornea can be.
Yeah, for that we have to first we have to confirm that autoimmune responses, that immune response is rejecting or accepting. For not only for cornea transplantation, all this transplantation, organ transplantation, first we have to check that acceptance.
Immune response.
Yeah, immune response, as well as that it may cause foreign substance like that. First we have to check that graft.
But probably if your research is extended further, it can be a good replacement for the cornea transplantation.
Yes,
Is that so?
because this one maximum acellular organ.
Yes.
Compared to other organs.
Okay.
That's why it's very easy to transplant to human to human.
Yes.
Nowadays, that pig cornea also.
Yeah, xenograft. Yeah, xenograft.
Xenograft also developing.
Okay.
Yeah.
But that's very impressive research. Yes. Thank you so much, sir.
Thank you.
So one last question. Good afternoon, sir. I just want to clarify, we all know that we have DOS layer, right? So it takes too long to wound healing for the process.
Yes.
So is that the nanoformulated therapy, it really works for the DOS layer?
Yes. Dual layer won't work.
Okay.
Because it is posterior layer.
Yeah.
It's not anterior layer.
Yeah.
When we connected with the epithelium, that stem cell based.
So it is limited to the epithelium and the basement layer of the epithelium.
Stem cell as well as epithelium.
Okay, sir.
Because that dual layer, it's intermediate.
Between the stroma and the descendant
Stoma, endothelium.
Okay.
Endothelium, it won't regenerate.
Okay.
That's why most of people in country, most of people suffered from different cataract and short vision, long vision like that. It's based upon the endothelial cell damage.
Okay.
That endothelial also it have only one layer compared to epithelium.
Yes.
Epithelium have multi-layer.
Okay.
That it instantly
It is a regenerative process.
But endothelium, it won't regenerate. When that at the birth time, how many cells are there until death? That epithelium, it stand constant.
Okay.
Intermediate our lifetime, that number of cell will die.
Okay.
Yeah. That cell structure also it modify. That's why we suffering so many issues.
Okay.
Still no one succeed for endothelial cell regenerations. That's the problem.
Thank you, sir. Thank you very much, sir. Let us once again appreciate our distinguished speaker with a big round of applause. And ladies and gentlemen, now it is a tea break. We just go for a tea break, take 20 minutes of tea break. After that, we will assemble for the validity session.
Minutes in 10.
tired of missing entries? तुम स्लो नहीं हो, बस सेटअप आउटडेटेड है. ग्रो का स्कैल्पर मोड ट्राई करो. बस एक्सप्लोर स्क्रीन पे जाओ और स्कैल्पर मोड ओपन करो.
Welcome back, ladies and gentlemen. We now begin the valedictory session of Elevate 2026. This session marks the successful completion of 3 days of academic excellence, international collaboration, and innovative research.
To formally welcome the gathering for the valedictory session, I request Dr. B. Bhuvanawam, Deputy Dean, ENT and HMS, to deliver a welcome address.
The most respected Chancellor, Honorable President Sir, esteemed Trustee Madam, Director Madam, Dean Sir, Chief Coordinators, JR Sir, Principals, distinguished keynote speakers, Head of the Departments, faculty members, research scholars, participants, and my dear students, a very warm and cordial welcome to all to the valedictory function of our conference. I extend my heartfelt appreciation to the organizing committee, faculty members, student volunteers, and all those who worked tirelessly behind the scenes to make this event a grand success. Over the course of this conference, we witnessed highly informative and thought-provoking paper presentations across various technical tracks conducted in both offline and online modes. The research contribution presented by the participants reflected innovation, academic excellence, and practical relevancies. The poster presentation across various disciplines were equally impressive, showcasing creative ideas, emerging technologies, and innovative research solutions. This presentation provided an excellent platform for the knowledge sharing and scholarly interaction. We were also privileged to gain valuable insights from our distinguished keynote speakers, whose excellence enriched our understanding of current trends, research advancement, and future opportunities in their respective field. Their inspiring addresses have undoubtedly motivated all of us to pursue excellence in research and innovation. With a great pleasure, I hereby declare the valedictory function open. Thank you.
Thank you, ma'am, for your warm welcome. Every successful conference leaves behind inspiring and valuable outcomes. To present a comprehensive report of the 3-day conference, I invite Dr. R. Mehta to present the conference report.
Respected members, our honorable resource person from India and abroad, respected faculty members, and my dear students, very good afternoon to everyone. It's my great pleasure to present the report of Elevate 2026, our 3-day international conference on SYNERGIZING artificial intelligence and the classical research in bioscience, engineering, and material science. As we gather for valedictory session, let us look back at the wonderful journey of the past 3 days. Day 1, July 10, 2026, our camp began with grand inaugural ceremony. After the invocation and Tamithal Vartha, we are welcomed by respected Dean Dr. B. Stalin Sir. The traditional lighting of lamp officially inaugurated the conference.
The conference chair, Dr. E. Shiba Precious, introduced us to the highlights of our university, and the conference convener, Dr. K. Sunadi, explained the vision and purpose of NV 2026. We were honored by inaugural addresses delivered by Dr. Mudarat bin Hali, University of Malaya, and Dr. Malade Batamila, International University. We also had inspiring felicitation addresses by Joint Registrars Dr. B. Tiruvalli and Dr. K. Saranan. One of the proud moment of the day was the release of conference proceedings, which reflects the valuable research presented at this conference. The technical sessions then began with excellent keynote lectures. Dr. Muhammad Hamdi Bin from the University of Malaya spoke about advanced nanostructured material for dye-sensitized solar cells. Dr. Malade Batamina from International University, Malaysia, delivered an informative lecture on machine learning in surface plasma reasons biodetections. Dr. J. Madhavan from Tiruvallur University spoke about bismuth-based semiconductor nanophotocatalyst for wastewater treatment. In the after, researchers and students actively participated in oral and poster presentation sessions within 9 tracks organized parallelly. The first day ended with a colorful cultural program followed by high tea, giving everyone an opportunity to interact and celebrate together. One of the outstanding highlights of the day was the drone intelligently developed by our Advanced Research Institute, which was showcased to peer researchers who had the opportunity to watch the developed drone in action. On day 1, the conference recorded a total of 1,012 registrations with 408 abstracts received. 168 poster presentation and 436 listeners on this. Day 2, July 11, 2026, the second day started with an inspiring lecture by Dr. Pulmurugan Karapaya, King Saud University, Saudi Arabia, on recent development in botanical and microbiological research. We were also privileged to attend two online international sessions. Dr. Theo Feng Hen from the University of North Asia spoke about water and environmental engineering in the era of artificial intelligence. Dr. Heiko Hedberg from Lund University, Sweden, shared his insights on the role of artificial intelligence in precision medicine. These sessions showed how technology helps experts from different countries share their knowledge with us. The remaining part of the day was dedicated to oral and poster presentation by students and researchers. They confidently presented their research, answered questions, and exchanged ideas in the reality academic mode. Day 3, July 12, 2026, today, the final day of conference began with 2 excellent plenary lectures. Dr. V. Vijayakumar from Vellore Institute of Technology spoke about cross-coupling reactions in the synthesis of quinoline compounds. Dr. Ramachandran Samuel from King Saud University delivered an interesting lectures on nano-formulated biomaterial on carnival wound healing. He also delivered today's validity keynote address. We have now reached the validity session. Soon we will listen the validity address, hear the feedback from the participants, and honor the best paper and the poster presentation through the distribution of awards and certificates. During these 3 days, NV26 has successfully brought together artificial intelligence and classical research. We had the privilege of learning from distinguished experts from India, Malaysia, Saudi Arabia, and Sweden. We listened to outstanding plenary lectures and witnessed a research presentation by our students and scholars. We had participants and listeners from the following institutes such as International University, Kingside University, Vellore Institute of Technology, Karanla University, Hindustan University, Tiruvallur University, Sankara University, Chayar Arts and Science College, Axelam College, Sri Shanmuga College of Engineering and Technology, Abdul Hakim BKM Arts College, Satyabala University, Sriperambur, Adi Parashakti College of Engineering, MMES Women's Arts and Science College, Sri Balaji Chakkalingam Engineering College, Dr. MGR Chakkalingam Arts College, Dr. MGR Educational and Research Institute Main Campus Chennai, SRM University, Peri College of Arts and Science, Arinara Anna Government College Chennai, Sri Sai Lalitandara Medical College, ASS Medical College, Chaitanya Bharati Institute of Technology, MIT Pondicherry, Alabama University Canada, Florida University USA, Savita University Chennai, University Chittoor, Tamil Nadu Open University, Sikkim Sardar Patel University, Sri Manakulam Vinayagar Engineering College. This conference became a great success because of the support of our management, dignitaries, resource persons, conference chair, convener, organizing committee, faculty members, student volunteers, and every participants. On behalf of the organizing committee, I sincerely thank everyone for your valuable support, active participation, contribution in making Elevate 2026 a memorable and a successful international conference. An exciting announcement, the second edition of Elevate is expected to take place in July 2028. We look forward to another edition of excellence. Thank you. Have a wonderful day.
Thank you, ma'am, for presenting an excellent summary of the conference proceedings. And the most awaiting woman of today's program is a valedictory address. It is a honor to invite Dr. V. Vijaykumar sir, Professor, Department of Chemistry, School of Advanced Sciences, Vellore Institute of Technologies, to deliver a valedictory address.
So, good afternoon to all. It's really an honor to me to felicitate the program. Generally, the international conferences, it is just giving a platform to bring all the well-known researchers in the world to come to a single platform to share their knowledge, experience, and other things to the young researchers. In that way, the Elevate 2026 is a successful one, and it is organized by Dr. MGR Educational Research Institute jointly with INTI International University and Colleges, Malaysia. So in that aspect, I am thanking both the institutions for bringing the eminent professors, researchers into a single platform to share their knowledge and experiences to the youngsters. So really thank to them. And in this aspect, I have to thank the Chancellor, Dr. A.C. Shanmugam of this institute, and Trustee Tirumati S. Lalita Lakshmi Madam, and President ACS Arun Kumar Sir. And I have to thank Tirumati Nirmala Arun Kumar, Director, Seba Ferix, Chief Coordinator, and Thiru A.C. Ravi, Secretary of this institute. And I have to appreciate the effort of Professor B. Stalin, Dean, and both the Registrar, Dr. K. Saravanan and Dr. V. Peruvalidi, Joint Registers, and Principal Faculty of Physiotherapy, and the conveners of the institute, convener of this conference, Dr. R. Sumati and Selvi, Dr. R. Selvi. So I am thanking them for bringing the international speakers in a single platform. So when I am going through the keynote speakers, I have seen that there are professors from Malaysia, from the eastern side, as well as from the western side. We can say that from King Saud University, Saudi. So it has covered almost 4 or 5 nations. and nearly 1,100 registrations are there, more than 500 abstracts are here, and so it shows that the discussions are going on, it went well, and nearly more than 25 institutions are participated here, so I can say that in this aspect, the conference has achieved its goal. So I am wishing the MGR University and NT International University and colleges to go together in the future years also for this kind of platform. Thank you. Thank you so much.
Thank you, sir, for your inspiring and motivation words. Your address has encouraged all of us to continue pursuing excellence in research and innovation. Thank you, sir.
Every conference becomes meaningful through the experience of its participants. We now invite a few delegates and participants to share their valuable feedbacks and experience about Elevate 2026. I now invite, is there any volunteers? Your good name? Harish. So, from? So, Mr. Harish from VIT.
I am Harish, I am from VIT. So I would like to thank the organizing team because as a chemist, to getting a collaborator from the interdisciplinary fields is very rare. In this conference, I got many collaboration from these peoples, from the international professors and more peoples, and so I would like to thank the organizer team very much. I gained so much knowledge from the presentation and from the plenary lectures because I'm now only starting my PhD journey, so gaining knowledge is very crucial for in these times. So very much thank you to the organizing team. Thank you.
Thank you, Harish. And now I call Ms. Gunaselvi S. from Hindustan Institution.
Good afternoon, one and all present here. I'm Gunaselvi, final year BTech Biotechnology student from Hindustan Institute of Technology and Science, Chennai. I'm really grateful for the opportunity to participate in this conference. It was a wonderful learning experience that allowed me to gain valuable knowledge, interact with researchers, and explore innovative ideas. The technical presentations were informative, well organized, and inspired us with the latest advancements in various fields. The cultural events were vibrant, enjoyable, and created a welcoming atmosphere for all participants. The overall organization of the conference was excellent, with every session conducted in a systematic and professional manner. The hospitality provided throughout the event was exceptional. The hostel accommodation was comfortable, and the care, support, and guidance extended by the organizing team made us feel safe, valued, and at home. Their dedication, coordination, and attention to every detail ensured that the conference was both memorable and enriching. I sincerely thank the organizing committee for their outstanding effort and wish them continued success in organizing many more such events. Thank you all.
Thank you, Gunaselvi, for your good feedback. And I will call Dr. Akbar sir, Assistant Professor, Faculty of Physiotherapy from Dr. MGR of campus.
A very good afternoon to one and all present here. It's my great privilege to give feedback on this 3 days international conference, Elevate 2026, which was organized by the Dr. MGR University, Aranya off campus. I found this conference were highly informative and well organized, and it's truly rewarding. And the keynote session were given by the expertise from various regions of the world, and the scientific session and interactive discussions were provided great insights, and it was very knowledgeable. And my sincere appreciation to the entire organizing committee from the university for well planning and coordination. It was well planned actually. And okay, so my heartfelt congratulations to whoever contributed to this conference to make it very great success. Thank you one and all. Thank you.
Thank you so much, sir.
Yeah, sure. Your name? So here Rangarajan from VIT to deliver the feedback.
Good afternoon all. I am Rangarajan, research scholar under the guidance of Dr. Kalaivanan, SSL VIT Vellore. So I am the tree, seed was made here. So I would like to express my heartfelt gratitude to the organizing committee for conducting such a well-organized conference. I want to sincerely appreciate their hospitality. It extended throughout this event till now. So the conference provided a valuable platform to gain knowledge from multidisciplinary perspectives. The key, mainly I want to mention the keynote session, research presentation and discussions. So I would like to proudly mention that I am the alumnus of Dr. MGR Chokkalingam Arts College, which is the constitution institution under the ACS group of institution. It was a matter of great pride to witness such an academically vibrant event associated with my alma mater. So I am very proud to be here. I would also like to express my gratitude to non-human with a human-like brain, AI, because without AI we are not here. So I want to thank that. So I also want to thank VITians who is working here as an assistant professors for a valuable insights and for asking questions and bothering me with their questions during my presentation. Thank you so much, ma'am. Colleagues who engaged me a lot during the conference session. The conference reflects institutional commitment to promoting the research. We really appreciate that, ma'am, and that's it. I'm not a person to congratulate. My sincere, last but not the least, my sincere appreciation to the management, organizing committee, faculty member, volunteers, and everyone who are the reason for this conference. Thank you so much.
Thank you, sir. Recognition is the greatest motivation for excellence. It is now time to honor the outstanding research contributions presented during this conference. My request, esteemed dignitaries, to kindly distribute the Best Oral Presentation Awards and Best Poster Presentation Awards. The list goes here.
The Best Orientation Award in the track 1, Miss Indu C. from Vellore Institute of Technology, the first prize. And the second prize goes to Dr. M. Sendil Kumar, 2VM College of Education. Okay. On behalf of Dr. M. Sendil Kumar, hitting Dr. Srivar sir from TVM College of Education 3. Next I will call Mr. Rangarajan T. from track 3, 4 and 5, got first prize in the oral presentation from VIT. Next I will call Mr. Harish P. from VIT who got a third prize in the track of 3, 4, 5 and 8. Next I will call Ms. Madhumita N. from Hindustan Institute of Technology and Science from track 7B. She got as an oral presentation. And next I will call Ms. Gunachelvi S. from Hindustan Institute of Technology and Science. She got a Distinguished Paper Award. And for poster presentation, I will call Mr. Sachin Krishna from VIT Vellore. She got first prize in the cadre of HNS. Congratulations to all the award recipients. I'll call all the winners to come for the group photo. Dr. S. Shivakumar. I'll call all the winners to come forward to the stage for the group photo. So let us appreciate them with a big round of applause for all the winners.
As we approach the conclusion of this memorable event, it is time to express our heartfelt gratitude to everyone who contributed to the success of Elevate 2026. May I now invite Dr. S. Sudhakar Sir, Principal, Faculty of Physiotherapy, to deliver the vote of thanks. Sir, kindly address the gathering.
முதற்கண் அன்னை தமிழுக்கு வணக்கம். Good afternoon to all present here. காலத்தினார் செய்த நன்றி சிறிதினும் ஞானத்தின் மான பெரிது. This beautiful couple by Thiruvalluvar remind us even the smallest act of gratitude offered at the right time is greater than the whole world. They say a vote of thanks should be a short, I truly agree. But after a wonderful 3 days conference like Elevate 2026, so many hands and hearts have worked behind the scenes. So I request your patience for just 3 minutes, not 3 days, as I express our sincere gratitude to everyone who made this event a grand success. It's a great honor and privilege to deliver a vote of thanks on this memorable occasion marking the successful completion of 3 days international conference Elevate 2026 organized by Central Research Facility off campus Orani from 10th to 12th July 2026. First, I express my heartfelt gratitude to our Honorable Founder Chancellor Sir, Honorable President Sir, beloved Managing Trustee Madam, Vice Chancellor Madam, Registrar Sir, Secretary Sir for their constant guidance, encouragement, support in making this conference a grand success. I extend my sincere gratitude to all our distinguished keynote speakers, Dr. Muhammad Hamid bin Ali, Dr. Malati, Dr. J. Madhavan, Dr. Pun Murugan Karpaya, Dr. Theo Fang N, Dr. Heiko Herwal, Dr. V. Vijay Kumar and Dr. Ramachandran Samuel for sharing their valuable knowledge and inspiring all of us. Thank you, Madam and Sir. I extend my heartfelt gratitude to INTI International University, Malaysia for their valuable cooperation and partnership in Elevate 2026. Thank you very much, Madam. I would like to express my heartfelt gratitude to Dr. Sheba Persis, Professor, Joint Registrar, Chief Coordinator of Off-Campus for her constant encouragement and excellent coordination throughout the conference. Her meticulous planning and unwavering support played a vital role in the grand success of the Elevate 2026. Thank you, Madam. My sincere thanks to Dr. R. Sumati, Professor and Deputy Dean Research and Innovation, Convener, and Dr. Selvi, Convener of the conference, for her outstanding coordination, dedicated effort and commitment ensured the smooth execution of every session and made this international conference a memorable and successful event. Thank you, Madam. I sincerely thank our respected Dean of Campus, Dr. B. Stalin, for his untiring support, constant guidance, and continuous encouragement. His leadership and commitment inspired us throughout the conference. We are truly grateful for your valuable contribution in making Elevate 2026 a grand success. Thank you, sir. I extend my sincere gratitude to our Joint Registrar, Dr. V. Peruvaldi, sir, and Dr. K. Saravanan, sir, Principal Fatima Grace, Faculty of Pharmacy, Deputy Dean Dr. Bhavana, for their constant support, valuable guidance and encouragement. Their cooperation contributed to the successful organization of Elevate 2026. And I sincerely thank the heads of various departments of off-campus, various committee members, special officer, director, principals of ACS group of institutions for their valuable support, cooperation and encouragement throughout this conference. Thank you, sir. I would like to express my heartfelt thanks to our wonderful comparing team, Dr. Ashwini Priya and Ms. Tarini, for conducting the entire program with confidence, grace and professionalism. Thank you, Madam. I sincerely extend my thanks to Mr. Gautam, Physician Associate, for his dedicated support in coordinating with our esteemed keynote speaker. Thank you, Gautam. I extend my sincere appreciation to the Food Committee for providing delicious meal refreshments with great care and hospitality. My heartfelt thanks to the Transport Committee for ensuring safe and timely transportation for all guests and participation. A special word of thanks to our Administrative Coordinator, Mr. Hari, Digital Banner Designing Team, Mr. Manigandan, for arranging every requirement on time and supporting all conference activity. Thank you. I also my sincere thanks to our media team for capturing every memorable moment and sharing the success of this conference with everyone. My sincere appreciation goes to the system admins, Mr. Vijayanand and team, technical team and electrical department for providing uninterrupted audiovisual support, internal facilities and technical assistance throughout this event. Thank you. I extend my warm thanks to all the faculty members, cultural coordinators, physical educators, non-teaching staff, volunteers, NSS team delegates from various institutions. Your dedication, discipline, teamwork, enthusiasm made this conference a grand success. Finally, I thank every participant for your active involvement and valuable contribution. Your presence made Elevate 26 a truly successful international conference. யாரேனும் பெயரும் விடுபட்டிருந்தால் அவர்களுக்கும் எங்களுடைய நன்றி. மீண்டும் சந்திப்போம். Thank you everyone.
இதில் ஒருத்தர் பேர் மட்டும் விடுபட்டுச்சு. அவர் வந்து டாக்டர் எஸ் சுதாகர். On behalf of organizing committee and all of you of my faculty members, students, I thank Dr. S. Sudhagan for his support in segregating the tracks and to conduct this poster presentation and paper presentation very smoothly. Thank you, sir.
Finally, we move on to the conclusion of the conference. With this, the 3-day international conference, Elevate 2026, officially comes to a close. Over the past 3 days, we have witnessed inspiring lectures, innovative researches, and interdisciplinary collaborations and meaningful academic exchanges. We sincerely thank all our keynote speakers, dignitaries, delegates, researchers, faculty members, students, sponsors, volunteers, and organizing committee members for making this conference a grand success. As we conclude the conference, may I request everyone to kindly raise up for the national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ कलवंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगल दायक जय हे भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Thank you all. Before we disperse, we request all the dignitaries, keynote speakers, organizing committee members, and award recipients to kindly assemble on the stage for the official group photo.
One by
Now I will call all the female faculties come forward for the group photo, including the external participants. Female participants, kindly come for the group photo. Now I will call all the male faculties, please come forward, including the external participants. All organizing committee members.
ஹைட்டாக இருக்குங்க மேலே ஏறிடுங்க சார் மகேஷ் சார் மேலே ஏறிடுங்க வழி விட மாட்டுறாரு பார்த்து பார்த்து
Thank you everyone. We wish you all a safe journey home and hope to welcome you again. Thank you all.